Can everybody see these slides? Cool. All right, hello everyone, and welcome to our 7th session in the Modeling Biology by Domain seminar series. Today we are going to be covering the domain of cancer modeling. The motivation behind this series is to try to understand the different biological domains that can be covered by computational modeling, which really span all of biology. And we've been progressively moving from single scale systems to more complex systems that generally involve multiple scales of biology. Today we'll be covering cancer, and we also have 2 more sessions in August and September on metabolism and whole cell modeling coming up. So we hope you'll be able to join us for those as well. Let's just go quickly through our seminar format. The structure will be a short introduction on the biological domain of cancer, about 5 minutes. We'll then have our talks, which are 20 minutes long, and a group discussion about 25 minutes long. The goals for this seminar are to identify our common challenges in cancer modeling, share our personal experiences with modeling and also solutions we've come up with, brainstorm new solutions, and ultimately create a collaborative community of modelers so that we have peers we can go to for support with our modeling challenges. For our discussion portion, our guidelines are not too strict. Err on the side of commenting. We want to hear from you. Honest debate is encouraged. Just mute your microphone, please, when you're not talking so that everybody can be heard. All right, so today we're covering the domain of cancer, and cancer models represent aberrant biological processes which can be driven by genomic instability or diverse sets of mutations and inflammation. Many of you who are working in the cancer modeling domain probably already are familiar with this image of the hallmarks of cancer, which are different behaviors that result at the cellular and subcellular level due to cancerous mutations, which can be sustained proliferative signaling, evasion of growth suppressors, activation of proliferation and metastasis, replicative immortality, induced angiogenesis, and resistance to cell death. And the biological basis for these, including the dysregulation of multiple intracellular pathways and extracellular signaling of key processes, are typically modeled in our field. A major barrier to modeling cancer is the inherent heterogeneity of the disease. This heterogeneity is not only considered with respect to the cell type being studied, but also to the tissue of origin, the stage of the disease, for example, whether or not it has metastasized to a new tissue, the interacting cell types within the tumor microenvironment, and also patient-to-patient variability due to genetic and environmental factors. So you may have distinct mutations at any point within the subcellular networks at the genomic level that result in different phenotypes between patients or within tumors. And also you have to consider that you're modeling multiple cell types because you're considering not only the tumor, the cancerous cells, but also the healthy cells that are in the surrounding environment and vasculature. So this heterogeneity is an inherent challenge of cancer modeling, but it also leaves an opportunity for computational modelers in conjunction with the medical genomics community and high throughput experimental advancements to support personalized medicine. For example, base models of intracellular signaling networks could be adapted to include patient-specific mutations and the resulting phenotypes identified using high throughput DNA sequencing or proteomics technologies. And simulation of such models and exploration of the perturbations within the network could reveal the most effective nodes for treatment intervention or disease diagnosis. So today we had invited 2 speakers to discuss their research in the cancer modeling domain, Dr. Paola Vera-Licona and Dr. Dirk Fay. Dr. Dirk Fay may not be able to join us today, so we hope he'll be able to join us at a future session if he does not join the call today. So we'd like to welcome Dr. Vera-Licona. We're very excited to hear about your research today.
All right, thank you so much. Let me share screen then here. Can you see my slides? Yes, perfect. All right, so before starting the presentation, actually let's remove here these windows. this side. Oh gosh. Okay. All right. So the presentation I'll be doing today is a computational systems biology pipeline for the reversion of resistance to cisplatin in a triple negative breast cancer subtype. Before doing the presentation, I want to give a disclosure about my unusual looks today. So even though I have had no hair when I was on my 20s because it's just being a badass, today that's not the only reason. So fortunately, I'm actually myself right now a cancer patient. So this work actually is becoming even more meaningful to me if I was not already very passionate about the topic. I think as you know, we might say all of these as researchers, be very conscious about the possible impact that our research hopefully might have. And I think in every single grant that I have submitted since I've been assistant professor, it has always a paragraph stating that you know, chemotherapy and surgery as the ways of treating a lot of cancer types, and this particular one, triple negative breast cancer, for instance, is really not a solution that we should be only offering to patients. And it's really terrifying when then you are in that exact situation, when you are being told that these are the only two options that you have for treatment. So this work is even more meaningful to me nowadays. All right. And so I'll give a little outline of the presentation. First, I want to give a biological motivation of the problem. So first of all, what is triple negative breast cancer? And specifically this subtype that we are working with, which is called the Tandem Duplicator phenotype. And then what we want to achieve in there with this reversion of chemotherapy resistance. So that's the biological motivation. Now, while the approach, I'll be talking about the structure-based control approaches and how we are applying it to solve this problem. Underlying, when I will be presenting about the approach, I'll try to give a motivation of why did we choose to have this approach, which actually is not only because of this type of cancer subtype, but because we at the same time want to generate also software tools that other people can use to apply similar, in similar way our approach. And then will close with some additional work and future directions. So what is triple negative breast cancer? So triple negative breast cancer, essentially, it's about 20% of all breast cancers. It is unusual in the sense that it is characterized by what it is not rather than what it is. So in this subtype, usually you have the 3 markers for breast cancer to classify them. So estrogen receptor, HER2 protein, and progesterone receptor. And in the case of triple negative breast cancer, it's because these 3 markers happen to be negative. So they are put essentially in that same bucket. But as you can imagine, they are really then also a very heterogeneous subtype. Also, the current targeted breast cancer therapies that exist in that sense, since all of these markers are negative and these are precisely used for the targeted therapies, they are not effective for treating triple negative breast cancer. Therefore, the only treatments that you mostly offer to patients nowadays, starting line essentially is surgery and very aggressive chemotherapy. And so in that sense, the 5-year survival rates are very low for triple negative breast cancer. For example, for other types of breast cancer is about 93%, whereas for triple negative breast cancer is about 77%. So what is a particular characteristic about this subtype of called essentially the Tandem Duplicator phenotype? So as Veronica mentioned, I mean, cancers are essentially driven by all of these genomic instabilities. You can have duplications, deletions, in different kinds of patterns of insertions, reversions, etc. And you can essentially expect that to happen in cancers and in any type of cancer for that matter. Tandem duplicators, what they are essentially about is essentially the duplication of a section essentially of the genome that is copied essentially one next to the other one. And tandem duplication really happens again in many cancers. We can have them as well. But what is very interesting about in triple negative breast cancer is that they have been found essentially to be very prevalent. Actually, this is the precise work of our collaborators, Dr. Menji and Dr. Ed Liu. So in their lab, what they actually saw, they analyzed essentially about 2,700 different tumors across different kinds of cancers. And what they saw essentially was that particularly for cancers like triple negative breast cancer, ovarian cancer, these patterns were repeated across the entire genome, because it's a very different thing to have a tandem duplication just in some regions and actually having across the entire genome and actually having with a particular distribution of sizes. So here what you see in this slide here on the left side is for non-TDP, you would expect that maybe you have across the entire genome and you have certain tandem duplicators of particular size of the span size, but then they are going to be distributed all over the place. What is interesting about what they found in cancers like triple negative breast cancer is that actually there are some very specific sizes of distributions. So these essentially are like 10,000 bases or 230,000 bases or 1.7 megabases. And so, and then combinations of them, and really the other ones are very little, as you can see here in this graph of the spans essentially. The other characteristic that they saw is that, well, obviously they are going to have a TP53 mutation, many of them. But then, for example, for 10,000 bases, they saw that they have, for example, BRCA mutation in methylation, and so even though they found some patterns, they really were not able to find any particular genes or drivers essentially in that sense. Though, it's very interesting the fact that you have these characteristics across essentially the entire genome. And the fact that they are so enriched in particularly, I mean, we are talking about having tandem duplicator very enriched in triple negative breast cancer, 55%, in comparison, for example, what it would be expected of less than 6% in general in breast cancer, over 50% also in ovarian cancer. And that also the fact that in many other cancers, maybe they have some tandem duplications, but across the genome, they don't. And cases like that are, for example, pancreatic cancer, leukemia, some brain cancers. So in a way, this is a very, very characteristic thing about essentially this type of triple negative breast cancer in particular. So we are very interested about understanding essentially what are these drivers, and essentially how can we identify some targets for this type of breast cancer subtype. The other thing that they observed, actually, Menji and Ed Liu and collaborators, was that actually these cancers happen to be very sensitive to cisplatin chemotherapy. And whereas it is known in the literature, for example, that BRCA1 mutation, for example, makes patients essentially be more sensitive to platinum treatments, what they think, actually, Menji and Dr. Liu think, is that this has most to do about the TDP pattern rather than only the BRCA mutation itself. And so what Ed Liu and collaborators in his lab did essentially was this patient-derived xenograft preclinical trial. And in that, as I mentioned, they revealed this sensitivity to cisplatin resistance, both in vivo and in vitro. And so what they did essentially was this clinical trial. They did it actually not only with cisplatin, they did it with other chemotherapeutic agents. But for the moment, we are focusing only on the cisplatin, since there it happens to be very sensitive. So here you have in this line on the x-axis essentially the time. So when the 3 doses were applied, so this is T1, T2, up to T3, and then these doses are essentially given weekly, and post-treatment, so this is PD, is when the measurements that we have are being taken. So I'll be talking about that after. On the y-axis, we have the tumor size. So as you can see on the x-axis across the time, first we have the treatment done. Then we see that for some of these tumors, the same tumor, but then applying to different mice, in some cases, essentially was very sensitive. So it shrunk actually at the very beginning for all of the treated mice. And those that were not treated obviously grew. This is what is called the vehicle. Then it was treated again. We have an intermediate point in there. And then we see here at this point also that after a third round of treatment, for the case that was not treated, will continue essentially growing once the treatment had been stopped in here. So this is the graph that grows. So it's essentially treated one time with cisplatin and then let it rest. And then another case is where they continue to be treated without stopping it, but then eventually it had developed resistance. So while it was shrinking, then it started growing again. And then here we have a point on which after treatment, it continued decreasing. So the measurements that we have essentially are completely untreated, treated the first time when it had shrunk, and then treated when it had developed resistance, and treated when it continued being sensitive. And so what we see here then is that while at the beginning it shows to be very sensitive to cisplatin, well, eventually many of these are going to develop resistance. So what we want to try to do is to find ways for identifying targets so that we can essentially reverse this resistance. And so what we can see here already, and this is something that we are not going to address into this talk, is the fact that also as it was mentioned at the beginning of in the intro part, tumors are heterogeneous. So obviously there is a part on which some cells in the tumor were already resistant, but then there are others that have acquired resistance. So we will want to essentially focus onto that after we have done this first line of analysis. So what we want to do essentially, as we said, is identify combinations of cisplatin resistance reversion drivers. As I mentioned to you already, but there are no targeted treatments today. And for triple-negative breast cancer, really, as far as I can tell, up to several months, the state is that while there are no targeted treatments, a lot of the current clinical trials, more than 80% of them are actually combinations of existing drugs. And so it is more or less reasonable to think that it's going to be hard to find one magic, silver bullet. So probably we will need a combination of targets to identify elements of resistance reversion. So here I have a representation of what we are trying to achieve. So this is just the metaphorical representation of Waddington landscape. I think we have seen this, for example, in the presentation when we saw about stem cells. And so what we are trying to represent here is rather not in the sense of cell differentiation, but about the Kaufman point of view about cancer, right? In which cancer, indeed, it is a result partly of mutation and environment, and that in the normal case, essentially, cells are able essentially to just have a normal behavior, normal phenotype, and that ultimately cancer attractors are existing in the normal, I guess, in any of these landscapes, dynamic landscapes of the system, and that they are only reachable once there are changes in the environment or there are mutations. So on that perspective, we are seeing here, or trying to understand this perturbation first of treating with a cisplatin, but that some behavior is this undesired resistance that we have observing here, whereas some others go into rather the sensitive phenotype or sensitive attractor. And then there are others that are, of course, on the untreated. And so what we want to do is essentially to change from this initial state that is going to eventually end up into this resistance phenotype, for those that are essentially acquire resistance, to return it back into the untreated attractor, or to essentially do it into rather a sensitive phenotype, as we observed for the other tumors that were sensitive across the treatment. So this calls essentially for a control of dynamical systems approach. So we have essentially an initial state that might end up in an undesired state. So what are the targets or combination of targets that we should perturb in a signaling network so that we can actually change the initial state to another one that actually eventually goes into a desired attractor? So typically what you would do into a dynamical systems approach that all of you will be familiar. This is just one example. We have, for example, multi-omics profile data, some literature evidence, some experiments. We can build the network topology independently, or the systems dynamics, or the dynamical model actually, together with the reconstruction of the network. And then have essentially the dynamical model. Then after having the dynamical model, we can actually see which are the attractors, since we are going to associate phenotypes in there, and apply control theory and combine to identify combinations of targets. So now here comes essentially where is the part on which what we are also trying to do here, what I am also trying to achieve in here, which is this approach. I mean, is obviously extremely useful and it gives us a lot of details, but the truth is that to build a dynamical model that is obviously a large scale representative of the system is simply not possible in many instances, in most of the instances, because of the lack of detailed mechanistic parameters, right? And so with that in mind, and also wanting to develop tools that help to essentially have a first-hand approach, an unbiased one where we can use essentially all the multi-omics profile data. We might have maybe some information on idea of about part of the system that might be important, but if we want to have a first unbiased approach where we let all the data and the literature all encompassed together to speak up first, and then after doing that analysis, essentially trim down what are the variables that we want to do more detailed mechanistic modeling of, then we really don't have anything today. There is this extreme on which we have the static network, and then we have a lot of network theory over there, which is extremely useful. We can have a lot of statistical approaches, which are extremely useful as well. But there is this gigantic gap then to have this dynamical systems perspective if we don't have the dynamical model. So this is also what we are trying to achieve in our research group. What we are trying to do is to use the network topology, but try to use some of the dynamical systems, not only approaches, but actually control theory approaches, and without necessarily needing, or without needing the dynamical model, to make some inferences about what are those possible combinations of targets, essentially for, in this case, achieving resistance reversion to chemotherapy. And then from there, have all of these different concerted perturbations that we are looking for. So how do we do that? We do that by using essentially a structure-based control approach, and considering that we have, in our case, a biological system specifically for nonlinear dynamics. So structure-based control methods have for objective identifying, based solely on the structure of the network, elements that can drive a system towards a specified goal or phenotype. Structure-based control approaches have existed really since the 1970s. One of the first ones to have been proposed was structural controllability by Lee. That was created actually for linear systems. And it was only recently in 2013, actually starting one of the first works into this direction that has been proposed actually for nonlinear dynamics. And this was a set of 2 papers by Mochizuki on which they propose that in order to control the system, we need the feedback vertex set. And so I won't have time to obviously go into all of the details, or the details in general of the paper, but just to have for you to have an idea of what this work is about. Essentially, when you have, I guess in dynamical systems, we have this concept of determining nodes, right? It's essentially a subset of nodes of the system by which once we essentially, we want to determine the dynamics of the entire system is enough to essentially have the information about this specific subset. So what Mochizuki and collaborators did in this paper in 2013 was to show that actually the feedback vertex set is exactly that set of determining nodes. In graph theory, a feedback vertex set is a set of nodes in a graph that when you remove them, it removes essentially the cycle of every single, the cycle of the entire network. And so this is extremely nice. The parting point actually with Mochizuki was to have essentially, to prove that essentially to have a dynamical system in a very generic form, as you can see above with the differential equations, for example. The only thing that I guess the change that he did in the proof of this finding was just to make sure to separate essentially the variables in the system that were helping for the activation of the node. And then obviously you're going to have a term that is going to talk about essentially the, I guess the negative term for decay, the decay term. And the only thing that he moved what he did was to essentially use an equivalent system on which for all of those negative set loops, essentially the term was actually moving to that decay term. In that sense, the equivalent system is that the static graph is no longer going to have negative feedback loops. It's only going to have positives. This is the only, I guess, point that I want to make sure to say, because otherwise, when sometimes I discuss about this paper, I see that sometimes there is this confusion. So in this paper, this is essentially what they said. In order to identify these control targets, we need to do essentially simply this feedback vertex set control. Then later in 2017, there was another paper, the structure-based control of complex networks by Sanudo, Yang, and Albert, on which they actually said that, different to Mochizuki, that you actually need also to consider the source nodes, since they are also going to play a role essentially to change the dynamics of themselves. They need to play also a role into changing the control. So in that case, the control nodes for that case should not only be the FVS set, but in addition the source nodes. So these are 2 different perspectives depending on how do you want to see the source nodes, whether they independently control themselves or they are not relevant for the entire system. In Sanudo's paper, essentially they proved that through 2 existent dynamical models in segment polarity genes model, whereas some years later, Mochizuki and collaborators in this paper, Controlling Self-Fate Specification, they actually went to show that for this particular system in the Assyrian embryo, it was enough to modify, as the theory had said, these 5 nodes in the FVS in order to essentially change the self-fate specification. And they tested that experimentally. And so this is the approach that we want to use and essentially apply it to our case. The difference, and I'm going to go very fast onto this approach, the difference, all this is theory saying, okay, if you want to control the system, you just need to perturb the nodes on the FVS set, and maybe the source nodes, if you go by having source nodes in your system, as of Sanudo's approach. But it's not telling you exactly how are you going to perturb specifically all of these combinations. And And noting that normally you have, if you really want to start with these large, rather networks that are more or less encompassing all of the different changes you observe on your high throughput data and literature and so on, which is on the order of hundreds, then feedback vertex set is on the order at least of several dozens. So you cannot do simply combinations of them and again to try them also just by doing essentially all combinations. So how do you find that? So this is essentially the part that we are actually trying to solve that question simultaneously. So, okay, so in this approach, as we said, we are going to try to first, from multiomics data, literature evidence, and experiment, construct the network topology. And so for that first step, I'm not going to go into all of the details, but essentially we are parting from having for all of these different comparisons. So when the tumor is untreated, and then the first time that is treated, then when it develops resistance versus the untreated. And so throughout all of these pairwise comparisons in the clinical trial, we are going to use essentially RNA-seq data, attack-seq data, literature, and databases. So we are going to do a bottom-up approach to build this network. We are first going to identify transcription factors that are upstream of these differentially expressed genes across the different pairs. We are also going to consider, since we have whole genome sequencing, mutations, which are extremely important when we are trying to identify transcription factors. And when we are going to pull essentially signaling pathways that are essentially on which the transcription factors identified are part of, we are also going to have to keep in mind mutations because the pathways are going to change. So we are using several tools also like Reconnect, Ensemble Variant Effect Predictor, and so on, in order to construct our pathways. In our case, we decided to do actually databases that are maintained and curated. This is why we decided to use Transfact and Transpath, but it is, we are also anyway corroborating with other databases. And so using, for example, Dorothea, and then also comparing back with already existing maps for triple negative breast cancer, such as the Atlas of Cancer. So at the end, when we did this process of comparing across all of the different pairs of the clinical trial and then joining the network that we have gotten out of that, we obtained a network of 888 nodes and over 2,800 nodes, edges, I'm sorry. And we here just have a summary of what it was captured altogether. So over 120 nodes associated with cisplatin resistance, 40 genes that are known to be associated with cisplatin treatment, and so on. So all of this was just done essentially following the data rather than anything beforehand. Also here we see that in this heat map, we have it across the different states of the treatment in the clinical trial, is that it was able to actually capture all of these differences in the treatment, slightly with exception of one of the samples on the T2 resistant and sensitive, which seems that essentially there are not that many differences into those two samples. The next step in the approach is then now to simulate the system, since our objective again is to perturb the initial condition, and what we want to modify essentially is the target essentially is the attractors, which is essentially what this FVS control is specifically for. And so to do so, since we don't have a mathematical model, well, we cannot do essentially a simulation, so we'd rather estimate attractors. So we are actually making a whole use of this work that actually came up in 2018, Topological Estimation of Signal Flow in Complex Signaling Networks. In this work, Lee and Cho essentially proposed to have an approach similar to that in social influence network theory. And in that case, essentially propose ways on which we can essentially assign genetic function essentially to all of the nodes of the network, depending on the sign of the incoming edges to each one of the nodes of the network, also the basal state or the original basal state of the system, and also the particular measurements, in this case, high throughput that we might have. So we make use of that to do our simulations. And what we want to do essentially is to be able for each one of the experimental conditions, so the vehicle, untreated, the sensitive, and the resistant, to be able to map them into an attractor landscape, just like what we do when we have a dynamical systems approach, a classical one. Since we only have these essentially 3 experimental conditions with our replicates, essentially we have 9 out of those. We needed to generate more points, so we essentially generated over 100,000 just with several random initial conditions, so that we have them all mapped into this landscape. And then we use machine learning approach, in this case k-means, to actually identify the clusters on which each one of our original attractors that come from experimental data are. So in this case, the green one, which is the untreated, the purple, which is the sensitive, and the pink, that is the resistant. So now that we have essentially some trained data and some clusters where we can see essentially where essentially the different attractors might be localized in this estimation of the attractor landscape, we go to the next step, which is now use actually this control approach of Mochizuki. And so we use the feedback vertex set control and then our tool Oksana. So this was actually the tool we already essentially released it. It's been now almost 8 months, I guess, since we released it. And the paper is about to be published in the Bioinformatics Journal. I hope it's going to come out in the next 2-3 weeks already. And so you can actually already use Oksana, the tool that we have developed. We have developed this actually in Cytoscape. I had done before during my postdoc actually a first version of what we had called Oksana. In this case, we did an extension. We have implemented in there also the feedback vertex set control of Mochizuki. We have implemented the feedback control from Sanyud and Albert. We have still included what it was originally in Oksana, and we have included the signal flow analysis. Essentially, all the pipeline that I am showing you, in principle, you can run it here into this essentially user-friendly Cytoscape app. So we are doing that. Here is a screenshot essentially of how our tool looks like. You upload your network, you select the essentially in this network what approach do you want to use, so Oksana or the FC or the SFA analysis, and then you can obtain the different combinations of control targets. And so the next step, so this is one example of a feedback vertex set that we obtain for our network. So it's essentially one very large one of over 15 nodes. And so the next step is actually now what combinations specifically of perturbations in each one of the nodes should we apply. So I will go very fast on this. And so in order to do so, what we need to do essentially is screenings now. So we have several FES sets, and we don't know whether we should essentially knock out or knock in or leave unperturbed each one of these FES sets of nodes. So essentially for many of these, we have several hundreds of thousands of combinations. So that's what we are actually trying ourselves. And once we do a particular set of perturbations into one FES set node, the question is now this one is supposed to, now this new perturbation, this attractor, is supposed to fall into which one of these 3 clusters. So in order to assign that, we need to use another machine learning approach. So we are using, for instance, k-nearest neighbors. So through k-nearest neighbors, we are asking this perturbation. Once we do it, then we use SFA to do the simulation, and we obtain an attractor. This attractor is supposed to classify as what? And so following this approach, we are able to see actually changes and be able to classify into what of the 3 cluster our perturbations falls into. So now that we are able that we know that we can use KNN to do a classification, now what we want to say is, well, let's start by a resistant initial phenotype from the experimental data that we have. We take the attractor, we do a perturbation according to the FES set, and then we want for that to have moved from the resistant now to the either sensitive or to the untreated, in the case, for example, of believing that this is acquired resistance. And so what you see here into this map is exactly one example of that. We identify, for example, a set on which by downregulating CREB1, COX1, PAX5, and TWIST1, and doing upregulation of RBPJ, then we are able to move actually from the resistant to the sensitive, essentially, cluster. And this is just one example. We have several of those clusters, several of these combinations that we are testing. We actually look into the literature, and actually 4 of these 5 nodes actually are very well known and related to either cisplatin resistance, and actually the directions that we identify are actually also matching what it has been found in the literature. So it seems that the method is actually finding good answers. Now, as I mentioned, this is also done this is all done in the bulk tumor. We acknowledge that this is not the case for tumors. They are heterogeneous to begin with as cell populations in the tumor themselves, the tumor cells. And so in the work actually of Kim, with also some of the collaborators of Ed Liu in Jeff Chuang's lab, they actually did essentially a breakdown of these tumors, specifically TDP and the one that we are working with, as a matter of fact, and realized that indeed there are some subpopulations that are more or less resistant subclones to cisplatin treatment. So we also have the data, and we are also currently working on that. In that, the objective is slightly different. So we are not only wanting the entire population to move to certain particular attractor phenotype, but we want certain clones, for example, those that were with acquired resistance, to move back to before they acquire resistance, to regain again that sensitivity, or to make essentially resensitization. And so we're already also trying that. This is another example, and seeing that actually it does work. So additional work that we are doing in this type of fashion, as you see all of the approach that we are doing. One, as we said, is for chemotherapy resistance reversion, but one part that I'm also very passionate about is this more extreme idea, which is finding combinations of targets to rather achieve tumor reversion. That is, to actually regain some of the normal phenotype. So this is actually well known in acute myeloid leukemia. This is what is called essentially the differentiation therapy. But then it has also been observed both in vitro and in vivo in other types of cancer. Actually, one of them is actually triple negative breast cancer, not TDP, another type of, another subtype, specifically Claudin low. It has already been observed. So I'm very interested in applying this type of approach and concepts to actually understand what type of combinations can be actually used to change into these more normal phenotypes. This is work, obviously, I will not have time to present any of that today, but this is something that we are doing aligned with this. Now, in terms of the chemotherapy resistance reversion, the future direction, well, concluding this analysis, we plan to follow up with dynamical modeling. Our hope is that since we are narrowing down the system, we find the FES set nodes, and we are actually finding specific combinations, and now we can actually take those that seem to be the most promising and then actually do dynamical modeling. So it's not a substitution, but actually something that can help us to bridge the work. As we said in the study so far, we have focused on intracellular studies. So we want to plan to extend this analysis to consider population level genetic heterogeneity, and then also tumor microenvironment. Obviously, these studies are done in PDXs, so we cannot talk about the immune system, for example, around, but at least we can talk about the population level heterogeneity there in the microenvironment, essentially. And so for that, we plan to take an approach of multi-scale agent-based modeling. And another thing that we are doing in parallel is to do these analogous studies, but then also to enter into the picture a study with microRNAs, for example, which is a collaboration we have just recently now launched with Pedro Mendez lab. And so I guess I am happy to take any questions you might have. Before that, I just will do my acknowledgements. So a lot of this work is done by my MD PhD student, Lauren Marazzi. But a lot of the tools that we have talked about have been also thanks to the work of another graduate student that recently graduated, Maddie Gastonway. And the Oksana tool was developed originally by one of my former postdocs, Andrew Gaynor-Dewar. And then Lauren actually did all of the work on the control theory and SFA part. And then, of course, an acknowledgement to my collaborator, Edison Liu, who essentially is one providing all of the experimental part for these TDP TMBCs. Pooja Kumar, who is the postdoc with whom we work very closely on this project, and Francesca Menji, with whom we originally had started working onto this project as well. And I'll be happy to take then any questions.
Thank you so much, Dr. Vera-Licona, for your talk. This sounds like a really interesting perspective, and I think it gives us a nice tool to use in conjunction with mechanistic modeling, which we've heard a lot about in previous talks, like you said.
Right.
Would anybody in the audience have any questions? You're welcome to unmute yourself and ask directly. Okay, well, I'll get us started then in that case. So I know you mentioned that your plan is to narrow down, basically create some kind of sub-network based on the FBS set of nodes that you identify. Have you done any preliminary, have you looked at the data to determine whether any of the sets you have identified are reasonable to target with pharmacologic treatments, or if it would be relatively easy to?
Right. So, I mean, we are doing a lot of these things. So one very important thing is actually, for example, to look at the lethality of these targets. So this is something that we do all the time with every set that we are obtaining. We are actually trying to see whether there is any kind of synthetic lethality. We are using databases actually from Broad Institute, which is very helpful to do. And for each one of these combinations, we are also doing a lot of literature review. One example was this one that I have presented to you that does actually make sense. Now, in terms of the practical world, and maybe I did not mention that when I narrowed down, we had originally this FBS set that had over 15 nodes, and we narrowed it down to a combination of 4 nodes. Why are we obsessed with 4? It would be ideal to have, obviously, as little as possible amount of nodes to perturb. So we are with another collaborator, also at Jackson Lab, with Albert. He essentially has developed a CRISPR-Cas method to essentially create a cassette in order to perturb several genes simultaneously. And so while we could do several combinations, we are trying to narrow down up to 4. And so far we have been able to identify sets of subsets essentially of 4. And then there is a lot easier to actually look on the literature and see whether they do make sense, whether they are really possible to do, at least individually. And then the experimental part, well, that is going to be our collaborator, Albert Chen, who's essentially going to have to do actually the experimental part to test the combinations. And so this is work that we are doing currently to actually try to decide for good subsets to actually test experimentally.
Thank you so much. It sounds like you have good experimental collaborators that you're working with very actively. That's awesome. It looks like we have a question from Rajanikan.
Okay. Hi, this is Raj. Very nice talk. I enjoyed it very much. One question I have is, how do you deal with when incorrect, so to speak, or changes in the network structure? Because it's not clear that every one of these in lines that we're drawing in our things are actually operational in a given context. They may not even be applicable noise, false positives, right? And that's one, and the related question to that is clearly some of these interactions are of different timescales than others. And so are there ways to handle that difference explicitly when you're searching for perturbations without even getting into the dynamics? Are there ways to weight these in terms of something about the dynamical system that will inform how certain states may or may not be reachable? So those are my questions. Thank you.
Yes. So the first thing, obviously, this is the 1 million question, right? We know that we don't have, in the model we might have essentially false positives or false negatives. So we try to craft essentially the building of the network, not only to do it automatized. We do a lot of careful search, whether it's on the literature, we combine with databases. For mutations, even though we use these amazing tools like Reconnect, for example, we still go and try to separately, in addition to read the literature, to actually make sure about the interactions that, particularly when we remove or add due to mutations, for instance. But there is always a danger, and I don't presume that we have perfect networks, and we also acknowledge that they do have essentially contain some issues. So that's number one, one important problem. And so the only thing that we essentially have done in the past with a lot of experiments, actually, both with the network reconstruction and actually with the use of this SFA approach for simulating the system, has been essentially to see to what degree this methodology withstand noise. And so well, in the case of SFA, for example, we know that they can withstand noise, but they can essentially get only accurate up to 65%. So we acknowledge that, and this is why, even though this is a very good first approach, I think, to have interesting targets, and I think, if we find combinations of targets that in addition individually in the literature look very promising, then we are trying to essentially go to the step of immediate experimental validation. But then this is why I'm also thinking that the next natural step is to actually do dynamical modeling, to actually be able to further test ahead of time. Having said that, also, in terms of the what you were mentioning about the time scales, and this also relates to SFA a lot. So as per the approach that we are doing, that I have presented up to now, we are using SFA. So that means that we are not taking into consideration time scales whatsoever. It is possible to implement them just the same way that in SFA they are implemented. They can just be implemented slightly with some time delays. The issue is that we do not have that information as well. So we are parting from not doing any of those modifications. And in fact, I guess at some point we will be able to know it is not complicated actually to change in terms of some of the nodes where that information might be known, to implement those time delays, for example, in certain variables, since the dynamics essentially of each one of the nodes is just modeled in this, I guess, approach that we have on SFA, just a mathematical equation, just follow it. So it can be slightly modified. So it's not complicated. It's just the lack of information that we don't necessarily have, particularly at this scale of hundreds of nodes.
If I may follow up on the issue of structural noise, right? Yeah. So if you were to build this, I mean, I'll accommodate from a slightly different angle, not from a methodological, but from a implementation angle, if you will, right? Yeah. Of different cancers. So if you take different cancers, it can't be that each one is driven by a completely different network of 5,000 node network, right? It can't be. There are not that many genes or transcription factors or key cell cycle regulators in the cell. So there must be a huge overlap in the kinds of networks that you would build. And on the practical side of things, it will tell us that all these cancers have their own drivers, their own intervention points, and each one is different and one doesn't work in the other. So that means that the structural noise in any given thing, which means that they're slightly different from each other, does matter in the end in terms of intervention.
Absolutely.
So that makes it, so if you take that knowledge and then turn back into the methodological aspect of it and say, what kind of a structural noise should we be evaluating for robust results? Then it may not be random knockouts and random bootstrapping. It might have to be a bit more guided by what we see. So just thinking about multiple cancers might tell us how we should think about structural noise, so to speak, and how the effect of it on the robust results.
Right. So I have to say, I slightly disagree with your statement that when you change, even for triple negative breast cancer, which in this case, the project that we are doing on tumor reversion, that I mentioned that we are working with a model essentially of Claudine low triple negative breast cancer. The network, I mean, there is some overlap, of course, but as you said, there, there's a natural overlap, but the networks are not necessarily the same. And a lot of the things change actually, again, because we are taking into consideration the mutations of the particular tumor of a study and so on. So there is not that large of an overlap. So it does matter. The only thing that I can tell you that we also have started to do essentially to try to gain some confidence. Let me put it like that, because you are saying that how can you gain some confidence essentially that the network that you are building actually the targets that you're obtaining make any sense, and the noise is essentially the structural noise is affecting or not. So one of the things that we are trying to do, for instance, is to try to see to what degree other other combinations of nodes that have similar connectivity patterns in combination, how much are they able to perturb the system to do this same game of moving from cluster of resistant attractors to others. So we are, essentially trying to identify several sets of those random sets that have similar connectivity pattern, and to see to what degree they are able to do those same changes, so that these FBS sets are not only randomly built and actually are meaningful into the network. So normally, if the structure of the network is close to or as correct as possible, these FBS nodes should be doing what they are supposed to do, since they are determining nodes. And so, but if not, then there is an issue, and the issue should be blamed to the structure of the network. So this is something that we are doing is a slightly side of actually knowing, because we don't know underlyingly, what are the changes essentially that we might have. In terms of actually doing experiments such as, well, what if we remove some edges and we see how these things change? We don't need to do that. Actually, that has already been done in both methodologies for structuralists. If you read the paper of Mochizuki, they have already say essentially and seen that when the FBS nodes are not doing what they are supposed to do, they are not really determining nodes, it is because of failure into the structure of the network characterization. And the same experiments already, if you see the paper of signal flow analysis, they also did the same type of experiments. They did change randomly several edges in the network, and then they saw essentially how the methods were able to withhold in that sense. So doing the experiments, we don't, because we know that it matters. And what we are doing is just simply trying to test with these randomly generated sets of nodes that have similar connectivity patterns, and then try to see whether the FBS essentially does not look just like any of the random sets. And that is going to give you some confidence, I think, also not on the FBS, because I mean this is the mathematical theory. I don't judge, I don't put it into question, but more about the structure of the network, because if not, then we have to revisit the network. I guess this is the closer answer that I can give you of what we have thought so far.
Thank you.
All right, I think we should probably wrap up, but thank you, Raj, for joining the discussion, and thank you, Dr. Vera-Licona, for presenting and for contributing to really good discussion as well. Looking forward to seeing the Oksana paper when it comes out soon.
Perfect. Well, you can already see it in bioRxiv.
Okay, awesome.
All right. Okay, thank you very much.
Thank you, everyone.
Bye.